All right, then let's talk about objective number 6.2, which is protein synthesis. So it says, state that a polypeptide is coded for by a gene, and that a gene is a sequence of nucleotides that forms part of the DNA molecule, right? Then state that a gene mutation is a change in the sequence of nucleotides that may result in an altered polypeptide. Describe the way in which the nucleotide sequences codes for amino acid sequence in a polypeptide with reference to nucleotide sequence in hemoglobin A, which is normal, and hemoglobin S, which is sickle cell. Alleles for the genes for the beta globin polypeptide. Describe how the information in DNA is used during transcription and translation to construct polypeptides, including the role of messenger RNA and transfer RNA, as well as the ribosomes. So let's now talk about the whole process of protein synthesis. All right, then let's talk about DNA and protein synthesis. So DNA controls protein synthesis. So we know that DNA is actually a single molecule, but it is said that it controls all the activities of the cell. So how is it possible? It is simply possible because just related to all the chemical reactions that are taking place in the cell, the metabolic activities, all of these are controlled by enzymes, and enzymes are proteins, right? So what DNA does, DNA is a code for proteins, right? And it controls which proteins are made, right? So specific proteins have to be made for specific functions, or specific enzymes have to catalyze specific reactions, right? So this actually is coded for by DNA. So DNA could just think of it as a dictator, right? So thus DNA controls the cell's activities, right? Now these protein molecules are made up of strings of amino acids. So these amino acids, right, will serve as monomers for these protein molecules now, right? So these, as the DNA is coding for proteins, so we will look at how these, like nitrogenous bases, adenine, guanine, cytosine, and how are they actually working, right? So these protein molecules are made up of strings of amino acids, right? The shape and behavior of a protein molecule depends on the exact sequence of these amino acids, right? That is, its primary structure basically of the protein, right? The primary basic structure of protein is actually what is decided by this exact sequence or exact order of these amino acids. So DNA controls protein structure by determining the exact order in which amino acids join together when a protein is made in a cell. So you can relate to it from a discussion in chapter number 2 also. So let's talk about gene now. So gene is a segment of DNA, right? So we have DNA, right? And in DNA, gene is a segment, right? And this segment of DNA can be transcribed and translated into a protein. So these are the two processes when we talk about protein synthesis. So these are the two processes that take place there. So one process, the one step is transcription, the second is translation. So this process takes place in the nucleus, right, where messenger RNA is formed, right? And then basic protein synthesis actually then takes place there in the cytoplasm, right? So this gene is a unit of heredity and the smallest functional unit of a DNA, right? So the smallest functional unit of the DNA is gene, right? So what happens is, as we're using the term transcribe, it is transcribed, yeah, or DNA is transcribed, right? So what happens is this gene or this segment of DNA, right? It is first changed to messenger RNA, right? It is changed to messenger RNA. So this process takes place in the nucleus. And then it converts into a polypeptide chain. This process takes place in the cytoplasm, right? So it is a segment of DNA that actually leads to the formation of a product, right? So whatever product is being made is a protein, so it can be an enzyme, it can be a hormone, right? So whatever product has to be made, right? So this gene actually leads to the formation of a product. Any change in the sequence of a gene leads to deformity in the product, right? So we'll look at how the sequence can change. Changes in the gene sequence can lead to mutations, right? So the product can become deformed or there can be mutations, right? And this can lead to serious genetic problems and diseases as well, right? So what is a mutation? A mutation that occurs in the gene is called a genetic mutation or gene mutation. Okay, so a mutation that occurs in the gene. Gene mutation is a genetic disorder that passes from one generation to the next, right? So this much about the gene. So just remember, it is the DNA. Okay, so gene is a segment of DNA. Okay, so it first it is transcribed into mRNA, right? And then it actually helps, it takes actually protein synthesis takes place because of that, right? So here they've shown you another version. So gene, and here we are getting a protein. All right, so let's start with protein synthesis. So the first process of protein synthesis is known as transcription, right? And the second process is translation. So transcription is a process in which messenger RNA or mRNA is formed from DNA template, right? So the process is known as transcription. Okay, so it's the formation of messenger RNA from a DNA template, right? This process takes place in the nucleus of the cell, and transcription requires a DNA template and an RNA polymerase. So we need an enzyme. The enzyme is RNA polymerase, and the DNA template has to be there, right? So here they have shown you this DNA strand, right? And it has been opened up, right? And we need the enzyme RNA polymerase. So we need two things here: DNA template, only one strand, and RNA polymerase. So the process of transcription is divided into three stages. Initiation. Initiation is definitely the process when it starts. Elongation, as the chain will actually will form to a certain length, and then termination. Termination is like when the process will stop, when whole of this messenger RNA has been transcribed, right? So transcription is the first step in the protein synthesis, right? So this much about transcription, and now let's talk about that in detail. So what is happening in transcription? In the nucleus, a part of the DNA molecule unwinds and unzips, right, as the hydrogen bond between the bases breaks. So this process is exactly the same as we have just dealt with in DNA replication, right? So it's part of a DNA molecule that would unwind and unzip. And remember, these are nitrogenous bases, so they have shown you one strand, right? And this is another strand of DNA. So it actually has opened up, right? It unwinds and unzips, and now these nitrogenous bases are exposed, right? And hydrogen bonds actually have been broken. Now, there are free activated RNA nucleotides. So again, activated RNA nucleotides are the one which have two extra phosphate groups attached to them. So again, you can relate it to what we have just learned in DNA replication, right? There we were talking about free activated, they were DNA nucleotides, right? So there are free activated RNA nucleotides now, like as we're talking about protein synthesis, they would pair up with the exposed bases of one strand only. This is what you need to remember, right? So if this is one strand here, so what you're looking at, this is strand here, this is actually this messenger RNA that is being formed, right? And it actually gets links up with only one strand. So these were the exposed nitrogenous bases. So it actually pairs up with this. As the RNA nucleotides pair up with their complementary ones, the sugar phosphate groups are bonded together by RNA polymerase. So up here in this case, the enzyme is RNA polymerase, and it forms a sugar phosphate backbone, right? So this is enzyme now, right? So as these nucleotides pair up with their complementary ones, the sugar phosphate groups are bonded together by RNA polymerase to form a sugar phosphate backbone. The new single-stranded molecule which has formed is called the messenger RNA, and then it leaves the nucleus via a pore in the nuclear envelope. So you know, in the nuclear membrane, there are nuclear pores, and through that the mRNA can leave the nucleus and enter into the cytoplasm. So here, I want you to show you this. See, so, okay, and remember, like in this case, if you're looking at this part here, look at these codings there, like complementary bases. See, since in RNA, there is no thymine, okay, but there will be uracil in its place. So adenine would pair up with uracil. So here you can see here, the complementary base. Adenine with uracil, adenine uracil, okay, guanine cytosine same. So there is a thymine in DNA, okay, but then adenine, guanine cytosine, cytosine guanine, okay, and then here you also see there was adenine, so there is uracil, right? So in place of in the RNA strand, okay, there will be no thymine, there are uracils here, right? And this one here, as they're looking, showing you here, this one has been formed and is leaving. Like these one are still like being like a pairing up, right? But as they will form, right? So as they will actually, they will have this complementary base pairs will be joined together, and this single-stranded molecule will then, when it is formed, okay, it will become messenger RNA and it will leave. Okay, so this is what is known as transcription. Okay, this is what is transcription. All right then. So let's talk about the role of this enzyme, RNA polymerase. Okay, so RNA polymerase plays an important role in the messenger RNA synthesis during transcription, right? So what happens at the beginning of transcription, RNA polymerase would attach to the DNA helix. So we just saw that in the previous diagram, right? Unwinding of DNA strands takes place and one strand is transcribed by the RNA polymerase, leading to the formation of messenger RNA from the DNA template, right? So we saw that in the previous diagram also, right? So here this is the RNA polymerase, right? So it would attach to this DNA strand and it will actually like this process of transcription of mRNA will take place. Now RNA polymerase then adds nucleotides that are complementary to the nucleotides on the DNA template, right? So these are the activated nucleotides for RNA. Now T on the DNA strand is replaced by a U. So we know there is no thymine in RNA. So that would be, if there was a T in DNA, it is replaced by U on RNA strand. RNA polymerase runs on the DNA template and an mRNA strand is formed, which is then released after termination of the transcription, right? After the formation of messenger RNA, it is released in the nucleus, okay, where it is then passes through the nuclear membrane, through the nuclear pore, and then enters the cytoplasm of the cell, right? So this is the role of RNA polymerase, right? And it happens during transcription. So here you can look at another picture. See, so here they have shown you a portion of DNA that has opened up, right? And this is the messenger RNA. So there will be free RNA nucleotides there in the nucleus, right? And as they are activated, they would start binding with this complementary bases along the DNA strand. So this will be done through this RNA polymerase, right? And then these sugar phosphate bonds will be made, right? So this will all happen because of RNA polymerase, right? And then you can see adenine is binding with uracil. Remember, it's RNA, right? So this thymine, it belongs to the DNA strand, right? So adenine, thymine, adenine, right? But in this case, adenine, there is uracil. So once this whole strand is formed, it will move out of the nucleus and it will enter the cytoplasm, right? So that was the process of transcription. Now let's talk a little about the triplet code. Now the thing is, this sequence of nucleotide bases, like sequence means, for example, cytosine first, then adenine, then thymine, right? So sequence of nucleotide bases in a DNA molecule is a code for the sequence of amino acids in a polypeptide, right? So in a polypeptide, which is this large protein chain, right? So it is made of amino acids which act as monomers, right? So which amino acid will link up with which amino acid, right? In the sequence or in which order, okay? This is all dictated by the DNA, right? DNA molecule. It actually, the sequence of these bases is actually the code. So this code is a three-lettered or triplet, or we call it codon. Okay, so these three letters are actually read as one code, right? So if it is CAA, so this will be one code. TTT will be one code. So it is read from here, left to right, right? Each sequence of three bases stands for one amino acid. So one amino acid is formed by these three sequences, right? So it stands for one amino acid. The sequence is always read in the same direction and from only one of the two strands of DNA molecule, right? So these are two strands. So you can see here, so it's 5' to 3'. Okay, so it is read in this direction, 5' to 3'. And this is the strand which is read. And the strand which is read, we call it as sense strand, right? So sequence is always read in the same direction and from only one of the two strands of DNA, and we call it as a sense strand. In this case, assume this is the lower strand of the diagram that we're looking. Complementary strand जो होता है, it is referred to as antisense. हम इसको antisense बोलेंगे, ठीक है? So reading from left hand end of the lower strand in the figure, the code is CAA. So this CAA codes for valine, right? So these are the amino acids, right? TTT codes for lysine. So these three, they will code for lysine. Then GAA, they code for leucine, right? Then CCC, they code for glycine. Now you do not need to remember these names, right? So there's an appendix at the end of your book also. So we'll see that it actually, there's a whole template made, and there we can find out that if this is a sequence of amino acids, so what, this is the sequence of these, sequence of these nucleotide bases basically, right? So this is they will code for this particular type of amino acid, right? So this short piece of DNA, so this is an example from your book. So this actually, it shows that these are 4 different, actually, it makes a chain of amino acid, and it is made for valine, lysine, leucine, and glycine, right? So this short piece of DNA carries the instruction to the cell. So it will say that make a chain of amino acid, and this is the sequence. So valine will come first. The next amino acid that will link up will be lysine. Then we will have leucine, and then we will have glycine, right? So again, for each particular protein, okay, there's a different like sequence of amino acids. And this is what is dictated by the DNA. So this is what we call as a triplet code, right? Okay. Now let's talk about the second part of protein synthesis, which is translation. Now, in the second stage of protein synthesis, what is happening? The messenger RNA that was formed there in the nucleus and has now entered the cytoplasm, it will be converted into protein, right? So it occurs in the cytoplasm of the cell. So translation also has 2 steps. Again, initiation. So the process starts, elongation as actually the strand forms, and then the termination when it stops. So translation we need many molecules, and the molecules that we need is messenger RNA. So definitely, like it was formed in the nucleus, it is in there in the cytoplasm. We need the ribosome, right? So ATS ribosome there, if we are talking about the eukaryotic cells, then transfer RNA, right? So let's talk a little about the role of ribosomes in translation. So ribosomes, you know, they have a larger subunit and a smaller subunit, and they play a role in translation. So for messenger RNA to be translated, the messenger RNA has to attach to the ribosomal subunits, and it generally attaches to the smaller subunit, right? So it attaches to ribosomal subunits, and then what happens? Transfer RNA, which is also there in the cytoplasm, during the initiation process of translation, right? So this actually transfer RNA will also be there, right? So this whole process, ribosome, so transfer RNA, it will come towards the larger subunit, right? So transfer RNA has the appropriate amino acid attached to it. So this transfer RNA is actually hairpin-like structure. We'll look at the diagram in the next slide, right? So it has one attachment site for the amino acid, and at one end it has these actually anticodons, right? Which are actually complementary to these messenger RNA. Like messenger RNA में अगर codon है, right? Those 3 triplets there. ठीक है? तो यहाँ पे transfer RNA में there will be anticodon. Right? Ribosome then scans the whole messenger RNA, and the subsequent transfer RNA adds the amino acid during the elongation process. So Abhi, just take it as a review as we are discussing the diagram, so it will become more clear to you, right? Then the ribosome dissociates from the messenger RNA only after completion of translation, when it reaches the stop codon on the messenger RNA. So we'll talk about that also. What is the stop codon, right? And what is the role of transfer RNA translation? Transfer RNA is required for incorporation of the amino acid corresponding to codon on the mRNA. Because we're saying it's protein synthesis is taking place. Protein synthesis के लिए we need amino acids. Okay, so amino acids are actually being carried by this transfer RNA. Okay, so transfer RNA is actually bringing them towards the ribosome. So transfer RNA has an amino acid acceptor site where it accepts the amino acid. It also has an anticodon site. तो उसके अंदर 2 attachment sites होती है. ठीक है, anticodon site that binds to the codon on the messenger RNA, right? The anticodon of transfer RNA is complementary to the codon on the messenger RNA, right? So this was just an overview. It will all become clear as we talk about these one by one. Okay then, so we have already talked about transcription, right? The messenger RNA was made. It came out of the nuclear pore, right? And it's now in the cytoplasm. What you see in the cytoplasm, these amino acids are there, the free amino acids in the cytoplasm, as well as their free transfer RNA. So see, transfer RNA actually is a hairpin-like structure. Okay? So this is one attachment site for amino acid. Okay? And these are what we call as anticodons, right? Messenger RNA has codons, right? The triplet code there that we just talked about earlier. So these are the anticodons. So it has 2 attachment sites. Okay? So each transfer RNA, okay, will have these specific anticodons. It will bind to specific amino acid, right? So this is what they have shown you as a transfer RNA, and here they have shown you like this is anticodon, right? And like for example, this transfer RNA has combined or has actually attached to methionine, right? So it has an attachment site specific of methionine. So only methionine will actually come and actually join this, right? So anticodon is which are UAC, right? Then this one, this transfer RNA, okay, it had this attachment site for tyrosine, right? And anticodon is AUG. So every transfer RNA will have a specific anticodons and will have a specific amino acid, right? So in the cytoplasm, there are free amino acids and transfer RNA molecules, as we just see in this diagram. At one end of each transfer RNA molecule is a site to which an amino acid can bind. At the other end, there are 3 unpaired bases. The base triplet is known as anticodon. Each tRNA molecule bonds with a particular amino acid under the control of a specific enzyme and with the energy from ATP. So I hope it makes sense till here. Okay, so here they've shown you the structure of ribosome. Okay, we'll have like another detailed diagram later also, but here they have shown you this is the smaller subunit and this is the larger subunit, right? So messenger RNA actually here attaches itself with the smaller subunit here. Okay, and the transfer RNA, okay, so they have shown you transfer RNA, it comes and then it is there at the larger subunit. Okay, so now let's see what happens, like what is the role of ribosome now. So up again, this whole process taking place in the cytoplasm. Now also in the cytoplasm, the messenger RNA molecule attaches to the ribosome, right? And you know the ribosome, okay, they are made of ribosomal RNA or rRNA, okay, as well as protein. So ribosomes are made up of rRNA and protein, and they contain a small subunit that you are looking at here, and there is a larger subunit, right? Messenger RNA binds to the smaller subunit, as you can see here. Acha, what happens is, like 6 of these bases at a time are exposed to the larger subunit. So this is the larger subunit. So if you count 1, 2, 3, 4, 5, 6. So at one time, or one particular time, 6 nitrogenous bases will be exposed to the larger subunit, right? So what happens? The first 3 exposed bases or the codon, these are the exposed bases. Remember, this is mRNA, and these are the exposed bases, or these are codons. Okay, so again, we are looking at a set of 3. Codons is a set of 3 here. So first 3, okay, are always AUG, right? So this is, okay, this is the first 3 are always, if this is A, this is U, this is G, right? So these are the codons, right? So the anticodons would be for adenine, it will be uracil, okay? For uracil, it will be adenine. For guanine, it will be cytosine. So these are the anticodons, right? And these are the codons, right? So the first 3, okay, are always AUG. Or transfer RNA ke andar, okay, complementary anticodons, we just saw it is UAC, right? So what will happen again, as like these are the complementary base pairs, so they will make hydrogen bonds. So we know that cytosine and guanine make 3 hydrogen bonds, like adenine and uracil will form 2, right? Then again, uracil, adenine, they will form 2 hydrogen bonds. So hydrogen bonds will be made between them, and this tRNA molecule has the amino acid methionine attached to it. So the first one, the first, okay, the first codon was AUG, okay, uska complementary anticodon was UAC, okay, and the first one, okay, for transfer RNA, methionine, okay, is the amino acid that is attached to it, okay. So the first amino acid will be methionine, right? So these 3 are, the bonds are made here. Okay then. Now what happens? Now a second transfer RNA came. So initially we only saw there was one transfer RNA, right? Now we have 2. So this is transfer RNA 1 and this is transfer RNA 2. Now a second transfer RNA molecule bonds with the next 3 exposed bases. Remember we said there are 6 of them exposed. So 1, 2, 3, 4, 5, 6. Okay. So this is done. Okay. Methionine amino acid is there. Okay. Now tyrosine is coming, and now we have these 3. 1, 2, 3. They are also exposed to the larger subunit, right? So they will again have, they will pair up with their complementary codons there, right? And what will happen at the same time? Methionine and tyrosine, a peptide bond will be made between them, right? And the reaction is catalyzed by an enzyme which is known as peptidyl transferase, which is found in the small subunit of ribosome. So the enzyme is found here, right? So its name is peptidyl transferase. So this actually catalyzes the reaction, okay, and catalyzes the formation of this peptide bond, right? So remember, at one time there were 6 nitrogenous bases exposed, okay, but these codons, okay, they will pair up with Okay, anticodon, which are 3 number. The first one is always methionine. Okay, and the first codon is always AUG, right? And then a second transfer RNA comes and it has tyrosine attached to it, right? So now because of this, a peptide bond will be made, and the enzyme that would catalyze that is peptidyl transferase. Okay, what happens next is that the ribosome now moves along. So see, this part is done. This is your messenger RNA strand. It will now move. Okay, so it moves in this direction. So it now moves along. Okay, the ribosome now moves along the RNA. So okay, ribosome actually is moving. Okay, ribosome moves in this direction. So definitely the chain is like, because this is part is done, so it will move along this direction. So it keeps on moving as 3 and 3 are being done. So it will keep on moving like this, right? So what happens, because ribosome is actually, it is reading the next 3 bases on the ribosome, right? So what is happening? A third RNA, transfer RNA would come and it brings a third amino acid, which joins to the second one, and the first transfer RNA leaves. Because remember, at any one time, there will be 6 nucleotides or nitrogenous bases that would be exposed there to the larger subunits. So again, if you look at this 1, 2, 3, 4, 5, 6, right? So these 3 are done. Okay, so it will move forward, and this ribosome will move in this direction, right? So see, this transfer RNA has left. Right, it had this anticodon, like, so it left now. Now what happens, up see, one by one it keep on happening, and you can see the ribosome is actually keep moving in this direction, and now we have this chain of amino acids, right? So this actually is one protein made, right? So it's a polypeptide chain. It continues to grow until a stop codon is exposed on the ribosome, right? So generally on this mRNA strand, there is the last one is actually what is called as the stop codon. Right, the stop codon can be any of these. Okay, it can be UAA, it can be UAC or UGA. Right, so if this sequence comes, right, so no translation will take place. Right, so this is a stop codon. Ribosome will not move. Okay, it will stop right here. It will not move ahead of it. So yahan tak ho gaya, it is done. Okay, it will not move any forward. Right, so this is how it's done. And this is the protein polypeptide chain. Right. Okay, then let's talk about the role of a start and a stop codon, as we were using these terms, right? So a start codon, okay, definitely this process has to start. So the start codon is required for the binding of ribosomal subunits and the transfer RNA, right? So ribosomal subunits and the transfer RNA. So there has to be a start, as we actually got this process, the first one we did. Okay, so a start codon has to be there, this one. Okay, because the process has to start. Right? So start codon actually checks the accuracy of translation. It generally codes for methionine amino acid. So remember we said the start codon, when we were looking at this diagram, we said the start codon is AUG. Right? So this is what will start the process, and it has this amino acid methionine. Right? So it has, okay, the most common start codon is AUG, okay, which is read as methionine. Now, stop codons are required for terminating the process of translation. Of when this process has to stop. Okay, when whatever protein was to be made, it has been made, right? So process has to be terminated, right? So now we will use the term stop codon. So in this case, what happens? No transfer RNA corresponds to the stop codon. Okay, so the stop codon can be UAA, UAC, or UGA. So when this comes, there is no transfer RNA that would correspond to this. Thus, the polypeptide chain breaks at the stop codon, right? So this is the role of a start and a stop codon. Okay, now mutations can also occur here, right? So mutations are changes in the genetic materials, okay, like DNA, okay, but these mutations can then be transmitted to the future generations. So these mutations are actually then become like they are inherited then. Now major causes of mutations are, they could be caused during replication, okay, and segregation of DNA. So we saw, okay, when DNA was replicating, so we had these nitrogenous bases and they were actually pairing up with the complementary bases. So some error can be can take place at that stage, right? There can be chemical mutants like formaldehyde, pesticides, mustard gases, caffeine, food preservatives. So they actually, okay, can cause mutations. Then rays such as ultraviolet rays, gamma rays, they can also cause mutation, right? Okay then. So mutations, like these are the mutations that can happen. Now detail of this is actually included in your A2. Okay, but like A2 biology, but just for your understanding, let's just see how actually mutation takes place, or let's just talk about the terms. So one type of mutation is known as substitution. Substitution means it means that one or more of the bases are swapped. For example, let's see. See, we actually, if you remember, we did it like as like three set of three. So it's ATG, then it is CCT. So substitution, if you look at G, this guanine has been substituted by T, thymine. Okay, so if you look at this here, there is no guanine, right? So this is substitution. So one or more base has been substituted. ठीक है, उसको replace करके guanine की जगह thymine लग गया. So it will be read at ATT and CCT. Okay, second type of mutation is addition. Addition, again, the name itself tells you it's addition of one or more bases. So for example, so if you're reading it as ATG and CCT, so look what happens. ATG remains, but see the second part was starting at CCT, but it will be read now as ACC because it has to be read from left to right और हमेशा 3 केस में पढ़ ले, ठीक है? So it will be read as ACC और उसके बाद जो next sequence वो भी बिल्कुल change हो जाएगा. Okay, then comes deletion. Deletion means removal of one or more bases. So see, there was ATG and then CCT. So see, AT के बाद G नहीं है, ठीक है? तो now it will be read as ATC. It was ATG, now it is ATC. फिर CT के साथ definitely जो next in line होगा, that will be read. अच्छा, then duplication can also happen. Duplication means it is repeating of one or more bases. For example, again, if it is ATG and CCT, so you can see CC यहाँ पे double है, तो C यहाँ पे we have more of them. So now we have 4. तो again अब जब read करेंगे 3 करके, so ATG, now it will be CCC. ठीक है? So see, CCT के बजाय वो CCC हो गया. So it will be changed. Then another type of mutation is inversion. Inversion का मतलब होता है reversal of base sequence. Reversal of base sequence, like if you follow here, जो इन्होंने red में दिखाया है, TGCC. तो देखें जो CC यहाँ पे लास्ट में था वो पहले आ गया and then GT. ठीक है, so all of it is inversed. So again पूरा sequence change हो गया. ठीक है, then comes translocation. It is a movement of sequence of bases from one location in the genome to the other, and this can be a form of movement towards a different chromosome or within the same chromosome. Right? So इसकी details सारी आप पढ़ेंगे basically A2 में, ठीक है? But these type of mutations that can happen, ठीक है? Nitrogenous bases की जो sequence है basically, ठीक है? वहाँ पे अगर change आएगा, so it can lead to gene mutation. Okay, so one example that we have to discuss is about that is sickle cell anemia. ठीक है? Mutation की example that we have to discuss in our course AS में. So sickle cell anemia is actually a type of जहाँ पे जो problem आ रही है, that is a substitution mutation. Right? So sickle cell anemia is actually a type of anemia, okay? So that actually with basically the problem aa rahi hai, that is in the hemoglobin molecule. So hemoglobin is a red pigment in the red blood cells, okay? And we know that hemoglobin ka basic kaam kya hai? It carries oxygen, right? So hemoglobin molecule, okay, it is made up of 4 polypeptide chains. Again, like if you can actually refer to our discussion from the. So if you have actually, if you go through the video lesson from the chapter of biological molecules, where we talked about proteins. So remember from that hemoglobin is a protein, right? But it has 4 polypeptide chains. So you can see they have actually done color coding here. So these are 1, 2, 3, 4. 4 polypeptide chains, and it has heme group in the center. So the one that you're looking at here, the bluish colors, okay? So these are the heme groups in the center. And the heme groups actually contain iron, right? So we have 4 polypeptide chains. 2 of them are known as alpha chains, and the other 2 are known as beta chains, right? So we have alpha chains here. So this one is alpha, and this one is alpha chain, right? This one is beta chain, this one is beta chain. And all of these 4 have this heme group in the center. So this is a hemoglobin molecule, right? Okay, the gene which codes for amino acid sequence in the beta polypeptides, right? We are talking about the beta polypeptides, beta chain, right? It is not the same in everyone. In most people, the beta polypeptide begin with this amino acid sequence. So this is the amino acid sequence. So here I've actually just pasted this list of 20 amino acids, right? So these are the names. So generally their first 3 letters are actually written here, right? So this VAL stands for valine. So you can find the name here also. So it's valine. then HIS is histine. So you do not need to actually remember these names, right? But just to show okay, how actually, so it's valine, histine, then LEU is leucine, right? The one that you see here, right? Then THR is threonine, right? Then we have proline, right? Then this GLU is for glutamic acid, and then lysine. So this is the actual sequence that codes for hemoglobin A, right? And it's the normal allele for the gene. But in people who have sickle cell anemia, what will happen? There's a base sequence which is CTT. It is replaced by C80. So substitution takes place. And what happens now in this whole sequence, instead of glutamic acid here at this place, it is replaced by valine. And look at the entire sequence, it remains the same. So it's valine, histine, leucine, threonine, proline. So this was glutamic acid, right? And valine has been substituted in its place. This is also glutamic acid, this is lysine. So only one has been substituted, right? So because of that, right? Because of that, the person, and if like the person has both of the alleles, which have hemoglobin S there, right? So that would actually become, the person would have sickle cell anemia then. As we just discussed, this type of mutation is called substitution. In this case, the small difference in amino acid sequence results in the genetic disease, and individuals, an individual who will have 2 copies of hemoglobin S will have the disease sickle cell anemia. So you can see how the shape of the red blood cell looks like. So these people actually do not have this ability to actually, the oxygen binding capacity of this red blood cell is decreased there, right? So let's talk about the effect of gene mutations on polypeptides. Now the thing is, most mutations do not alter the polypeptide, right? So most of them do not alter, right? Or only alter it slightly, so that its appearance of function is not changed. So we look at that, okay, how it happens. However, a small number of mutations code for a significantly altered polypeptide with a different shape, right? This may affect the ability of the protein to perform its function. For example, if you again recall about enzymes, enzymes have an active site, right? And they have this specific substrate that would have the complementary shape to the active site, right? So if the shape of the active site of an enzyme changes, and remember enzyme is a protein, the substrate will no longer be able to bind to the active site. Okay? And if then like that enzyme substrate complex is not being formed, right? So whatever activity had to be taking place will not take place. Other than that, a structural protein like collagen, it may also lose its strength if its shape changes, right? So these are the effects of gene mutations on polypeptides. Now, just an example for you. A silent mutation is one in which the nucleotide sequence, there's a change in that, but it results in the same amino acid sequence. ठीक है, तो amino acid जो बनता है वो same ही होता है, हालांकि nucleotide sequence में change आ जाता है, ठीक है? So this is possible because some amino acids can be coded for by up to 4 different triplet codon sequences. We will look at the appendix in a while, that is in your book, ठीक है? So sometimes it also happens क्योंकि कुछ ऐसे होते हैं, कुछ amino acid they will be coded for by 4 different codons होंगे, but they will be coding for one single amino acid, right? So silent mutations are often a change in the second or third base in the codon, ठीक है? So हमारे पास 1, 2, 3 bases होते हैं, तो second या third में difference होगा, first में नहीं होगा, ठीक है? For example, valine is coded for by 4 different triplet codon sequences, ठीक है? So 4 codons उसके ये हैं, GUU, GUC, GUA, GUG, ठीक है? So see, first one is the same in all of them, ठीक है? So second या third के अंदर difference होगा. Therefore, as long as the first 2 nucleotides in the codon are guanine and uracil, valine will be inserted in the polypeptide. So just have a look at that index in your book. So we'll see that. So this is appendix 2, DNA and RNA triplet code. So this is actually a ready-made template for the code that you can simply use to identify which amino acid is there. So for example, like right now they said about valine, right? Valine is coded for, right? And they said the sequence could be, right? It could be GUU, GUC, GUA, and GUG. So let's just see that. Okay, so this would definitely be in the RNA portion. So let's just see that. Okay, so this is how we actually read it. So this is G, the first one. Okay, so this is the first position, the second, and the third, right? So we are looking for G, U, U, right? So first one is G, this is second, this is U. ठीक है, so G है, हमें उसके साथ यही वाला सामने वाला पोर्शन देखना पड़ेगा, ये वाला ब्लॉक. ठीक है, so this is U. ठीक है, and then third position में then again same block में you will look at U. ठीक है, so U from the second and U from the third. So see, it is coding for valine, right? अच्छा, what else they said? GUU के अलावा they said GUC, GUA. So let's just see that also. GUC देख लेते हैं. See, तो again G, second position में U और third position में here is C. ठीक है, अगर मैं ऊपर वाला erase करूँ तो see, here is C. तो again if you follow this, it again is valine. Right? So see, valine is being coded for actually like 4 different sequences, but they are coding for valine. So अगर यहाँ पे sequence change भी हो रहा है, तो you will have the same amino acid. Right? अच्छा, similarly, remember a while back they said that AUG is actually coding for the start. Okay? So there is methionine. So let's just see there. So if we are looking at AUG, so this is A, the first one. This is U, the second one. So we'll look at this block. Okay? And we will look at G in the third position here. So G is over there. Okay? And if I follow this line here, okay, you can use a ruler also. So see, this is methionine, right? All right then. Then we also talked about the stop codon, right? So for example, so here you can see, okay, so it says stop. So for example, if it was A, okay, T, and let's say T, A, T, T, okay, so A from the first, T from the second, and again T from the third, or ATC. So it will be the stop codon there, right? So I hope it makes sense. So these are the DNA and RNA triplet codes. Okay, so this is how these are read. And we are done with this chapter. And we'll start with the past papers and shown in the next class then. Take care.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.